Hello everyone and welcome to this special alumni event, Unlocking the Power of Pandemic Therapeutics with Offscript. I'm your host, Dr Matt Agnew. I would like to begin by acknowledging the traditional custodians of the land on which we meet this evening, the Wurundjeri people of the Kulin Nations, and we pay our respects to all elders past, present and emerging. While COVID-19 may feel like it's in the rearview mirror, leading infectious diseases scientists in Victoria are already preparing for what comes next. At the heart of that effort is one of Australia's most ambitious medical frontiers, pandemic therapeutics. Unlike vaccines, therapeutics are drugs that treat and in some cases prevent serious illness. They're well established in diseases like HIV, but not yet for the next wave of pandemics. That's about to change. At tonight's exclusive event hosted by the University of Melbourne and presented by the Cumming Global Centre for Pandemic Therapeutics, you'll hear directly from researchers on the front line as they reveal groundbreaking technologies being developed right here in Melbourne. Tools that could transform how we respond to global health threats. If we get this right, we can save lives, protect communities, and keep society moving in the inevitable event of the next pandemic. This event also marks the official launch of Offscript, the new podcast from the Cumming Global Centre for Pandemic Therapeutics, which I had the pleasure of hosting. This series features leading voices in medical science, including Federal Health Minister Mark Butler, Doherty Institute Director Professor Sharon Lewin, Australia's Chief Medical Officer Michael Kidd, and former WHO's Chief Scientist Jeremy Farrar. Speaking of our Federal Health Minister, Mark Butler, he was kind enough to record a video message in support of tonight's event, which we would like to play for you now.
Well, good evening, everyone. At the outset, I'd like to acknowledge the traditional owners of the land on which you're meeting, the Wurundjeri and Woiwurrung and Bunurong peoples of the Kulin Nation. I extend that respect to their elders past and present, and to First Nations people who are with you today. I'm recording this on the lands of the Ngunnawal people. I was delighted to take part in an interview for the Offscript podcast earlier this year and thoroughly enjoyed my conversation with Professor Sharon Lewin. When it comes to world-leading medical research, as you know, Australia has long punched above its weight, and it's a sector that continues to grow. Today, around $1.5 billion is invested by the Commonwealth Government in research every year. Our government is proud to invest in our world-class medical research sector to help our researchers transform their ideas into life-saving interventions. We have outstanding medical researchers right here in Australia whose idea can bring better health outcomes for Australians and also for people right around the world. We're delivering a number of important initiatives and programs to further strengthen the country's research capabilities, including, importantly, in the area of pandemic preparedness. The National Health and Medical Research Strategy is one step closer now, with consultation opening on a draft at the end of August. This landmark strategy will accelerate health and medical research in Australia to deliver innovative and equitable health outcomes. It will not only enable bold and transformative ideas, but also strengthen our global leadership in health and medical research. I encourage members of the Australian health and medical research community to review the draft and provide feedback to ensure it reflects your vision and your priorities. Our government has also introduced legislation now to deliver on our commitment to establish an independent Australian Centre for Disease Control. The Australian CDC will be an independent statutory agency that brings together critical information and experts to protect Australia from diseases and other threats to public health. Our government fully recognises the critical role also that therapeutics must play in preparing for the next pandemic, because we know the earlier we can deliver treatments, the better for the community. During COVID-19, we saw the enormous pressure placed on our hospital and aged care systems, and frankly, in aged care, that pressure continues. The ready availability of antiviral treatments for older Australians in aged care facility continues to save lives. Antiviral medications have also played a vital role in the HIV epidemic, now spanning more than 40 years. These treatments have transformed the lives of people living with HIV, who with this therapy can now live long and healthy lives. For hepatitis C, we now have a highly effective virus-clearing treatment on the PBS, something that seemed unimaginable just a decade ago. Just as critical as ongoing investment in foundational research is our collective effort to build and maintain trust in science. It's vital that the community has confidence that governments are acting on the best available advice, informed by the latest research. And that is why our government's collaboration with highly skilled researchers from the Cumming Global Centre, the Peter Doherty Institute, the broader Melbourne Biomedical Precinct, and right across the country is just so important. Thank you to all the researchers and broader scientific community, not only for keeping Australia at the forefront of global medical research, but for helping to keep our community safe. And congratulations to the Cumming Global Centre for Pandemic Therapeutics team on tonight's launch of the Off Script podcast. I'm really sure this podcast will be a catalyst for important dialogue on the value of therapeutics in pandemic preparedness. I encourage you all to tune in to these important discussions and like, subscribe and share it among your networks. Enjoy the evening.
An important message, and it's fantastic to have the support of the Federal Health Minister on an issue as critical as pandemic preparedness and the all-important role that therapeutics can and must play. Before I introduce my first speaker, a few housekeeping notes from me. Tonight's discussion is being recorded and will be available to watch via University of Melbourne channels after the event. And we would love to hear from you, and we'll have a small amount of time for questions at the end of the discussion. Many thanks to those who submitted questions when you registered, and we will do our absolute best to answer them, as many of them as we can. So I would now like to welcome Melbourne Laureate Professor Sharon Lewin to the stage. As many of you may know, Professor Lewin is the director of both the Peter Doherty Institute for Infection and Immunity and the Cumming Global Centre for Pandemic Therapeutics. She is a world-leading infectious disease expert driving research and innovation to improve pandemic preparedness and global health outcomes. Professor Lewin, of course, was one of my favourite guests to interview as part of the Off Script podcast, and I thought we would play a snippet of our conversation before diving into our chat here tonight.
So if we had access to that sort of antiviral therapeutic much earlier on, how a really effective therapeutic, how might that, how might our response to the COVID-19 pandemic have looked?
Oh, I think it could have looked very, very different. And in fact, we had done some modelling on millions of lives saved by access to an antiviral for a few reasons. First of all, it can sort of hold the fort, so to speak, while your vaccine's being developed. And remember, when you have a vaccine, it takes a few weeks to build up your immune response to the vaccine. You've got to have 2, maybe 3 shots. It takes a few weeks, sometimes months. So it can fill in that gap while you're developing the vaccine, administering it and waiting for people to develop their response to the vaccine. You still need antivirals even with an effective vaccine, because the people that got infected where the vaccine didn't work and had a high risk of going on to disease, they still need antiviral drugs. So it stops illness. And it's incredibly important if you can't develop a vaccine.
Off Script is brought to you by the Cumming Global Centre for Pandemic Therapeutics. Listen wherever you get your podcasts.
Okay, please welcome Professor Sharon Lewin. Sharon, welcome. Riveting conversation by the two of us, if I don't say so myself.
Looking a lot more casual.
Yes, I was actually. A little more cleanly cut as well. A couple of scene-setting questions to start with. Firstly, very big, broad question though. What is a therapeutic?
Well, a therapeutic is a fancy word for a drug or a treatment once you get infected with a virus. It could be a tablet, it could be an injection. A therapeutic can be given when you're well but infected and to stop you becoming sick, or it can be given once you are sick, for example, in hospital and need treatment. And people often get confused with, we all learnt what vaccines do for infectious diseases. They ideally prevent you getting infected, but you still need therapeutics to treat people if they're at risk of getting very sick and they become infected. And as I mentioned on the interview, on the question you asked me on the podcast, we could really be in a position one day where we have an outbreak of a major infectious disease that we cannot develop a vaccine for. And we've had experience with that in HIV. I've worked most of my career, last 30 years, in HIV, where we have no HIV vaccine, but you have fantastic antiviral treatments or therapeutics, and that's what's turned the HIV pandemic around. It's what Minister Butler was talking about. And he also used another fantastic example, which is hepatitis C, a liver disease for which we have no vaccine for, but have curative treatments for. So this is why treatments are so important for tackling infectious diseases.
So yeah, you mentioned exactly that. You've got such a comprehensive body of work behind therapeutics as they apply to HIV. How have they worked effectively so far with regards to HIV?
Well, the therapies that we use for HIV directly target the virus, and they stop the virus from replicating, and that's what an antiviral does, or what we call a direct-acting antiviral. They're the best things you could have. And 40 years ago, when treatments were evolving for HIV, smart chemists worked out how to stop HIV from replicating by targeting certain points in its viral life cycle. And we've used that sort of technology for hepatitis C drugs, for hepatitis B drugs, and even for COVID drugs. It's the same technology. And HIV drugs, they've been around for 30 years, but they're now going through a revolution right now, which I find fascinating. So up until when HIV drugs first came along in the mid-90s, people literally took 20, 25 tablets a day, 3 times a day, loads of side effects, very difficult to keep people on treatment. And then now, or 10 years ago, we got better and better and better, and now you just take literally a single tablet a day. And in the last 5 years, we've also developed injectable antivirals, so you can be on an injection every 2 months. And now the technology is getting even better, that maybe we will have treatments for HIV that are just a single injection every 6 months or even 12 months. So there's just and we may not even need a vaccine if we could really make antivirals broadly available.
Right. So even though sometimes it's referred to as sort of the second line of defence, it often can be the line of defence in some of these instances.
That's right. It might be your only line of defence in these very rare, viruses are incredibly clever. They're much cleverer than we are. HIV, hepatitis C, they've worked out how to persist, and our immune system can't get rid of them. Now, COVID was, of course, very, very different. COVID, making a vaccine was relatively easy. And very quick. We had vaccines, you'll remember, that were already rolled out, within 12 months of isolating a brand new virus, and they worked really well. I mean, they weren't perfect, but they worked really well. But antiviral drugs, which are just as important, took a lot longer for us to develop in COVID. We didn't really have very good oral antiviral drugs for at least the first 2 years. And yes, it's a second line of defence. It's like, you've got the fire department and you need a backup plan. And so really you need both. You need both anyway, but if you don't get a vaccine or if you need time for the vaccine to work, antivirals can fill that spot. I just want to add one other thing about HIV. So I talked about treatment. So people that have HIV, if they go onto antiviral drugs and take the drugs, people with HIV now have a normal life expectancy and can no longer transmit the virus. So it improves health and stops you from being infectious. But antivirals can also be used to prevent you becoming infected with HIV. So they can be used for prevention as well. Many of you will know when you travel to malaria, places with malaria, you can take an anti-malarial drug to prevent you getting malaria. Well, that also works for viruses. And that has revolutionised the HIV response. It's really the only virus where we use antivirals for prevention, but that could also play a role with a new pandemic.
Right. So yeah, obviously, what is a therapeutic? Not necessarily a very clean-cut question or answer to give to such a complex and large question. But with that understanding of what they are, now that we've had a bit of a deeper dive, let's get into why they're becoming a central pillar of pandemic preparedness. And so with that in mind, what is the Coming Global Centre for Pandemic Therapeutics and why was it developed?
Well, I think when you have a new outbreak, as we had with SARS-CoV-2, a totally new virus that we never understood or never knew before, you've only got a few things up your sleeve. There's public health measures, which were lockdowns, etc., which everyone in this room will remember very well, of course. Don't want to trigger any trauma, but you remember all that in Melbourne very well.
It's all shared.
The next tool we have is diagnostics, to be able to identify who's infected so we can separate the people infected from those that are uninfected. The third is a vaccine, and we did very well on developing vaccines because there were decades of investment in new vaccine technologies that preceded the pandemic, and a lot of investment in technologies that could rapidly adapt to a new virus. And that was brilliant foresight in the vaccine world. And the third is, and the fourth is therapeutics, is drugs that you could use, just like we use in HIV, to treat people who have the virus or even prevent people from getting the virus if you don't have a vaccine. And they took, in a pandemic like we had with COVID, with a respiratory virus, you don't have a lot of time up your sleeve to develop new drugs. Drug development takes years to A, identify the drug, B, prove it's safe, and 3, get it out into the community. And you don't have a lot of time in a pandemic where you need these tools very quickly. And so what this idea behind the Cumming Global Centre was to identify better technologies that will give us therapeutics at speed. If we use the technologies that have been used for HIV or for hepatitis C, which work extremely well, those drugs generally take years to develop. And every time you have a new virus, you need a new drug. So we want to work on technologies that could be adapted much quicker. And with a whole lot of new changes in science, the science of HIV drugs is 30, 40 years old. There are a whole lot of other new ways you can do this, and that's what the idea was for this centre. We also did, I referenced it when we chatted, we also did some modelling on how, first of all, how much. We looked globally on how much money had been spent on developing COVID vaccines compared to how much money had been spent on developing COVID therapeutics. And we did this study in October 2020. And we came up with a result of around $100 billion that had been spent on COVID vaccines and around $7 billion that had been spent on COVID drugs. So it's an area that has not been invested in, and we've also largely relied on pharmaceutical companies to develop new drugs. So what we want to do is develop new technologies that will then be picked up by pharmaceutical companies to develop the drug.
Right. And I mean, speaking of financing and donations, it would be remiss of me not to mention Jeff Cumming, where the namesake for the Cumming Global Institute comes from. Did you want to speak about the philanthropic donation from Jeff Cumming?
Yes, of course. So Jeff's gift was absolutely transformational in being able to develop and deliver on the centre. And actually the design of the centre was heavily influenced by Jeff's approach to business, Jeff's approach to philanthropy. So just by way of background, Jeff is a Canadian. He's a businessman, he's an economist, and he has moved to Melbourne at the time of the pandemic. And he was actually introduced to me by Jim Angus, who's sitting in the front row, previous Dean of Medicine, Faculty of Medicine, Dentistry and Health Science here at University of Melbourne. I'm just using Jeff's words. He said that he spent most of his life acquiring money, and he now wants to spend the rest of his life. He's now, I won't tell you how old he is, but he's now later in his life. He wants to spend the next 10 years of his life giving it away. He's very passionate about medical research. His father was a doctor in a small town in Canada, and he'd previously given $100 million to the University of Calgary, where he went to university. And so he was very rattled, as many business people were, as many politicians were, as many of us were, by the pandemic and how fragile things were and how very rapidly things seemed to unravel. not only, issues around what we had to do with children and going to school and businesses and all the things that we went through, and the huge economic costs to the world in the range of trillions of dollars, let alone the millions of people that died. So Jeff wanted to make a very significant gift to ensure that we would not be in the same position that we were in 2020 in 20 years' time. And when I first met him, that was on a rainy, dark night in July 2021 together with Jim. That was really wanted to do. He said he wanted to give a gift to humanity, and he wanted to make sure we would never be in the same position we were in 2020. And of course, there was lots of things we could have done. There's so many areas of pandemic preparedness that we need to do better to ensure we aren't in the same position as we were in 2020. But I did, on that first night, I did speak to him about what I thought the gaps were, and antivirals were top of my brain because of all of the experience I've had with HIV. But I did talk to him about better diagnostics, better clinical trials, better data collection. There were so many different things that we could have done better. And I think Jeff played a very important role in, as we talked, and many others were very involved in these discussions. Jim McCluskey, who was instrumental in setting up the Doherty Institute, was involved. Peter Doherty was involved. Then Vice-Chancellor Duncan Maskell. Jeff met many, many people. He really wanted us to do something really well at scale and incredibly focused. And I think he brought these principles of his success in business to how we might think about solving a major problem. He's not a scientist. He didn't really know what an antiviral was when I met him. In fact, on that first night, I thought he was going to sleep as I was describing it to him. So I thought, I've lost him on this one. He's not that interested in antivirals. But then he can now explain it to you, probably far better than I can. So Jeff was instrumental. He gave us the biggest gift in Australian history to medical research. That's $250 million over 20 years. Extraordinary gift. And the Victorian government have supported this because of their commitment to being better prepared for the next pandemic, but also at the huge potential of us in Victoria, not only developing better therapeutics, but all of the economic benefits we hope that will bring to the states. And the Victorian government also contributed $75 million over 10 years.
Very exciting. It's like you said, there was a huge speed at which we developed vaccines during the pandemic, and hopefully this is an accelerant for therapeutics to have a similar sort of rapid response in the future to avoid similar situations. Let's get a little futuristic. What's on the horizon? What innovations are getting you most excited right now?
Well, there's lots of science is changing at a very, very rapid pace now because of great technologies. So everyone knows about mRNA for vaccines, but mRNA, and you'll hear more about it from our expert Paula, who'll be up talking next, but mRNA is basically, and Paula will give a brilliant description, but mRNA can be used for so much more than just vaccines. It's essentially a way of giving instructions to your body to make something new that it hasn't made before. So we can use mRNA as therapeutics, and I think that is just a whole new avenue of research. Again, the Victorian government and really many other states in Australia invested heavily in mRNA technology at the beginning of the pandemic because mRNA vaccines were so important and Australia needs capabilities to make mRNA. And so we have a great opportunity here in Victoria because of the Victorian government having invested in not just bringing Moderna to Victoria and BioNTech, but also funding mRNA science. I'm really excited about mRNA, and Paula will tell you more about that. I think I also am very excited about tackling the genetic code of the virus rather than tackling the proteins that it produces. And the reason why that would be a very big advantage for therapeutics is because we know the genetic code of the virus very quickly. In fact, the genetic code of the virus was released on January the 10th, 2020. And so if we had things that such as gene scissors that can target the code of the virus, which is usually RNA or DNA, that could meet our criteria for developing therapeutics quickly. And then there's also amazing advances in just technologies to understand how the virus affects the body. And Ash will talk about this, but we can now understand what every cell does at a single cell level. And single cell analyses is giving us a lot of insights into the immune response. So they're just a couple of things that are, and I've chose those examples because one is a therapy, but one is also understanding the immune response. And part of the Cumming Centre is committed to what we call discovery science, so that to just understand how viruses replicate, how the immune system responds to it. That will deliver us the mRNA breakthroughs of the future. But if we don't understand the discovery, the real basics, we're not going to get the transformational therapeutics. And one thing that Jeff was very visionary in, it wasn't my idea. I'm used to 5-year cycles with grants and getting papers out and getting your next funding. He really wanted to fund this on a 20-year cycle. So he wants us to be doing the scientific discoveries that will deliver therapies in 20 years. That's not to say we take it easy. That's just to say we want to do something that will be transformational.
Fabulous. And just going to give you one quick one just to finish with, and it's going to be a bit unfair because it's not a quick question to answer. But what is if you had one takeaway message for your audience here tonight, what would it be?
My one takeaway is that science can do incredible things for the major problems we face, like pandemics. But science on its own is not good enough. So we do have to make sure that science is understood by the community so that people actually do trust the scientific solutions that we're giving to people. So the community has to come on the journey with us. And so it's both. It's science and the community engagement. Yeah.
Absolutely. Thank you for your time, Sharon. Let's all thank Sharon. Fantastic discussion. As I said, we will have some time for some Q&A at the very end, so please do hold on to your questions for Sharon for now. I would now like to welcome 3 people to the stage who could very well be working on Australia's next big medical breakthrough and who are at the front line of preparing us for the next pandemic. Dr. Paula Savall and Associate Professor Ash Hake from the Doherty Institute and Professor Wei Hong Tam from the Walter and Eliza Hall Institute of Medical Research are all Cumming Global Centre researchers, having received foundation grants for projects to develop novel therapeutics for pathogens of pandemic potential. And tonight we have the unique opportunity to look under the hood and hear from some of Melbourne's most innovative thinkers about their work. Please thank them all to the stage. You may have seen Dr. Paula Savall in global media in recent months, from The Guardian to the BBC and the New York Times. In partnership with Professor Sharon Lewin, she pioneered a new strategy to cure HIV using mRNA and gene editing therapies, which she is also applying to develop pandemic therapeutics. I also interviewed Paula for Off Script, and we had a fantastic and enlightening discussion. Before I jump into the questions I have for her tonight, I thought we would play a snippet of our conversation.
The COVID-19 pandemic was one of the first examples of where we used something like this, which we tend to call platform technologies. So it's essentially a therapeutic that's made up out of different Lego blocks that we put together. And so if the individual Lego blocks have been validated already, the combination of that and the final therapeutic can be processed through all the required regulatory and safety steps much more quickly because we already know its parts.
Right.
And I understand that has been something that maybe us scientists needed to communicate better to the general public. And I think we're going to see more of these vaccines or more of these therapeutics go through those regulatory processes and be tested in the clinic. And I hope that at least over time we can explain this a bit better. And I feel that we scientists owe that to the general public.
Off Script is brought to you by the Cumming Global Centre for Pandemic Therapeutics. Listen wherever you get your podcasts.
Again, another a riveting conversation, if I don't mind saying so myself. Paula, you've used an analogy, and I mentioned this during our chat as well, because I really do like it. An analogy in the past that likens mRNA technology to a mail delivery driver delivering a message to ourselves. Can you explain that again to me and the audience?
Yeah, of course, not a problem. I think it's a good analogy because it explains what technology is, it explains what it does, and it explains some of the challenges that we have with using it in a therapeutic setting. So the mRNA really is like a recipe that can give instructions, in this case, to a cell to do something. It's like a recipe that instructs you to cook a different cake or to cook a beautiful Indian dish. So we can use the same format, the recipe, to give instructions for loads of different things. So it could be the instructions that help us build a vaccine, but these instructions could also be the next therapeutic. And the issue, though, with this mRNA or this recipe is that it's very fragile. It's like you have a recipe on a medieval parchment. You know, it's like it's really fragile. You need to protect it, and so we need to encapsulate it in a box, and that box needs a delivery man to really deliver the recipe to the right address, to the right cell in the body, so that the cell can act on the recipe, can act on the instructions, and do what we want it to do.
I love the analogy because it just breaks down so succinctly and clearly. We've spoke on it, obviously in our chat, but obviously tonight we've touched on it, and Sharon has as well. But you've been using mRNA, or your work on using mRNA platform to develop a cure for HIV and also for respiratory pathogens has been discussed and touched on a couple of times. Can you talk us through that a little bit?
Yeah, I think it's similar to what we touched on previously and what Sharon also mentioned. So I also used to work on HIV, and we were trying to develop curative therapeutics that were better than the already amazing medications that Sharon described. So we currently have these direct antiviral treatments that suppress the virus, but you need to take these for the rest of your life if you are living with HIV. And so a lot of what we do in our laboratory is try and develop a next generation of therapeutics that you only need to take for maybe a couple of months, after which you can stop taking all treatments and the virus will not come back. And as part of those efforts, we started working with this mRNA technology. And like many others, in the medical field, we realized that even though HIV and COVID-19 or the next pandemic all sound like completely different medical challenges, and in a way they're completely different viruses, but we realized that a lot of the fundamental technologies that we use to either study them or to try and build therapeutics to treat those viruses are very similar. And so it was relatively easy to pivot from working on HIV to using the same mRNA and the same delivery technology and try and apply that to develop the next generation of COVID therapeutics or for the new pandemic.
And speaking of that a little bit, just to kind of wrap things up a little, what would you say, and again, it's probably a hard question to articulate because there's several steps, but the critical next steps to translate your research directly into a patient-ready therapeutic for the next pandemic?
I think the biggest challenge in terms of developing a pandemic therapeutic that is based on this mRNA and the delivery vehicle, we call this a lipid nanoparticle in complicated terminology, is that during COVID-19, we developed this technology as part of a vaccine. So what we needed our recipe to instruct is your immune system. We needed to tell your immune system, hey, this is what COVID looks like. If you see this again, please start a fight. And so we did that by delivering the mRNA into your arm through an injection, which most of us have had here in the room. That meant that the mRNA reached cells in our arm, eventually went into a lymph node where it had its effect. If we're trying to pivot this technology and instead develop a therapeutic, which Sharon so beautifully described, is something that we might need to take when we're already ill. If this is the next respiratory pathogen, so something that infects our airways and our lungs, we don't need this therapeutic in our arm, we need it in our lungs. And so we need to change the way we deliver. The mRNA is the same, but we need to change the way we deliver it and where we deliver it to. So what we're trying to develop is a way to develop inhaled, so a way to almost like an asthma puffer, and that you will inhale the lipid nanoparticles with the mRNA so that they directly reach the lung and can treat disease.
Thanks, Paul. Very, very exciting stuff. I'd like to move down the line now to Associate Professor Ash Haig. You might recognise Ash from Medicine or Myth, the SBS program he co-hosted alongside Dr. Charlie Teo and Dr. Ginny Mansberg, where they decided which home remedies were worth testing in clinical trials. Today, Ash brings that same curiosity and clinical insight to his work at the Cumming Global Centre for Pandemic Therapeutics. He studies how lung cells respond to viral infections using a cutting-edge technology called spatial transcriptomics. That's a mouthful, which maps gene activity and cell locations to help prevent severe disease in future respiratory pandemics. Ash, before we jump into your latest work, I have to ask, the wackiest remedy you came across on Myth or Medicine, Medicine or Myth, and did any of them actually make it into mainstream medicine?
Thanks for asking about that, Matt.
Came in for the serious questions.
There's three of them. One was I tried Reiki for the first time,
Okay.
which is where someone holds their hands very close to you, but doesn't touch you, and uses their aura to help you. That was very relaxing.
Yep.
One was dried barbecued seahorse for prevention or treatment of asthma.
Okay.
I don't think that won anything.
Okay.
And there was another one called Miracle Berry, which was by a remarkable couple who'd set up a farm in Queensland, growing mangosteens, but then grew these tiny berries that accentuated the taste of certain products. And so it really, they pitched it as potentially helping people who have dysgeusia, like bad taste after maybe cancer treatment. And I tried it, and it works.
Okay.
So that's the one thing.
That's the medicine and
that was the best one.
The other is the myths.
Yeah.
Okay.
The other one's the best. So.
Yeah. Okay. Enough fun with the myths, though. Let's get back to the medicine. Let's talk about spatial transcriptomics. Am I getting that right?
Yeah, good.
Spatial transcriptomics. For those of us who aren't molecular biologists, I think I'm speaking for a number of people in the room, including myself, what exactly does it mean, and how are you applying it in your research?
So I'll try a different analogy to Paula's. 350 years ago, people started looking at biological samples with a microscope, and the shape of a cell, or the way it's positioned within a tissue, can be really informative. And we still use that today and depend on that in our pathology labs to look at biopsies that people have had, and they need to learn about their health condition. So it's critical science, the microscope. But just imagine, rather than a microscope, if you said you could actually ask that cell a questionnaire, and not a questionnaire of what cell are you, which you can tell by looking at that cell, but you could ask all the nuances of what are you doing right now, and what are you doing with that cell that's next to you? All of these things essentially are encoded in the message that we talked about before that cells use. So there may be as many as 20,000 different messages going on within a cell. We could count them, and we can, based on that 20,000 list, start to get some inferences and guesses around what's going well, what's going badly wrong with that cell or its neighbours.
Right. And so how does developing that better understanding of cells and how they're interacting with other cells and within the lungs, how does that help develop better treatments for respiratory diseases?
Well, you'll know that the response that any given human body has to one virus, unfortunately, is different. And there are some of us who had an infection and we almost never knew about it. And there are other people in our community who became very, very ill indeed and needed hospitalization and treatment. All of that difference is based on how your body responds to that infection. And at a microscopic level, it's dictated by how one cell may respond poorly and how it might interact with another cell. If you can start to understand the way that cells use whatever language to interact with each other, well, maybe you can intervene. If you can, in a sense, decipher that code, you can form strategies for intervening.
Right, and going on a slightly different angle here now, discussing why in particular respiratory viruses are among the biggest threats in terms of future pandemics, because they do spread so rapidly. This work that you're doing, if it's successful, how could it help us respond more effectively in the case of the next respiratory-based pandemic?
Well, the technology you mentioned that's unpronounceable is very useful, because not only can it take a human tissue sample and tell you what the cells are doing well and what they're doing poorly, that they can actually even detect the virus. So you could get in one single sample what's going well, what's going badly, what is it that's doing it, where is it, and all of that information could be vital in maybe helping that individual, but if not, because that individual, God forbid, might have passed away, well, that information will be critical for helping the rest of the people in the community. Right.
Fantastic. Thank you so much, Ash, for providing a deep insight into the research you're doing and spatial transcriptomics. I'd now like to move on down the line again to Professor Wei Hong, who joins us tonight from the Walter and Eliza Hall Institute of Medical Research. She also works with the Cumming Global Centre for Pandemic Therapeutics, where she develops rapid nanobody platforms to create therapies against pathogens of pandemic potential. Wei Hong, nanobodies are only found in camels, alpacas, llamas, and wobbegong sharks. How does that work?
I know, it's such a crazy quirk of evolution, and it was found, a discovery found about 30 years ago, that alpacas, llamas, and actually the wobbegong shark have evolved this really cool antibody entity. And I think Sharon talked about it, when you get a vaccine, you have antibodies. Antibodies, in some cases, are the first line of defense of your immune system. But these animals have just made through evolution this very weird entity, which is the smallest antibody entity that you can get in the natural world.
So, pardon my reductive point here. So nanobodies are basically very small antibodies, which are commonplace in medical treatments. So what benefits do nanobodies bring that antibodies can't?
Well, so this is a nanobody. In your system, you don't have this. This is a single domain antibody, so it exists as a single module. In your bodies, you would have this paired with another chain. They call them heavy and light chain. The beauty of this is that it exists as a single domain entity. And so naturally, this is the smallest protein that can actually bind the thing that you want to target. So if you can think of this as a design module, it then becomes very modular and very easy to link to other antibodies. And so in a lot of therapeutics, it's sometimes beneficial to deliver two antibodies or two different drugs so that you actually reduce the potential of developing resistance. And so actually having something that exists alone and you can design very easily with another partner, and if I could get Ash to maybe help me out here. This becomes a very, very powerful design principle in terms of therapeutic development.
And what would it mean if we had shelf-ready nanobodies ready to deploy, ready to go when the next pandemic hits? What would that mean? What would be the implications?
The implication is preventing disease. And so, as Sharon mentioned, vaccines are amazing. They're an amazing tool, but they require an immune response to occur, and that can take a couple of weeks. If you had an antibody therapy, you can deliver that, and those antibodies are ready to actually fight infection, and so they work straight away. And so Minister Butler referred to the aged care system and how infection is still going through and what happened during COVID, as most of you are aware. If we had an antiviral antibody therapy at that point, we could have actually ring-fenced that infection. So we could actually deliver it to people in aged care and stop that infection straight away. And so what we mean by ready to roll off the shelf is understanding enough of the biology, understanding how to design something to a new virus and actually being able to take that and actually stopping infection straight away. So I think it's very powerful. It's a wonderful complement to vaccines that do require a bit more of a time for immune system. And so the pair of them together, I think, is going to be the thing that's going to get us out of the next pandemic.
Fantastic. Thank you so much, Wei Hong, as well as Paula and Ash. We appreciate, obviously, all your insights you've shared with us tonight, as well as the research that you do and the passion that drives it. I'd now like to ask our speakers 2 final questions before opening up for Q&A with the audience. So I'm going to hit you with both the questions, and then we'll go down and you can go through them. So what makes platform technologies so exciting in the development of pandemic therapeutics? And with all the innovation happening right here in Melbourne, do you feel optimistic about our ability to manage the next pandemic? So I'll start with you, Sharon. Oh, we'll start down the other end. Yeah, we'll go
so
down
we have to answer
in reverse
both
order.
Of them.
To the best of your ability. I won't be. Yeah.
I mean, for us, platform technologies are fun because they make a very efficient system, right? And so Sharon spoke about this development at speed. And so a combination of understanding the science, but then being able to build a system that's adaptable to another disease that you see, that's what we call platform technology. You make the system so efficient that any mail parcel could kind of go through. And the second question, because you've asked it so nicely.
Yeah, with all the innovation happening here in Melbourne, do you feel optimistic about our ability to manage the next pandemic?
I am actually very optimistic because we saw this happen through COVID. We saw the collaborations that were occurring not just in Victoria, but nationwide. We didn't just see the collaborations with the scientists. We saw the collaborations with, government. We saw the collaboration with state governments, within our precinct, with the clinicians as well. And I think one of the biggest ethos of the Cumming Centre, and we hear this all the time from Sharon and Jeff Cumming, is collaborate, collaborate, collaborate so that we can move these things faster. So I do think that we have that ethos around us in terms of infectious diseases. We do know in the infectious diseases field, the next pandemic will occur. It's not a matter of when it will occur. And I think, building from this idea and building from this, the Center of Disease Control and people understanding the science as well and us being able to communicate more. But I think as a community, we are a lot more collaborative through COVID and having the bridging of the platform technologies pulling us together in such a way and this wonderful call to build at speed, I do think we're going to face the pandemic in a much more positive way.
Ash, did you want to weigh in?
Was that a pun? You used her name.
It wasn't intended that way, but...
Shocking. Platform technologies for us, it provides us with an ability to be nimble. So we can do our own research in a particular area, and my group is focused on malaria. But it allows us to collaborate with our colleagues and to work on viral infections, to work on bacterial infections, to work on cancer. So that's also another aspect of, that facilitates collaboration. In terms of am I optimistic? I am, not only from the perspective of increased collaboration and fostering that, but here in Victoria, the pandemic has, I think, encouraged bodies like mRNA Victoria. It's encouraged huge investment from people like BioNTech and Moderna. But I think the single biggest thing that I'm most optimistic about is it's forcing and encouraging us to communicate with our community. And I think that's really, really critical. And it's that really powers a lot of us. How are we going to make ourselves both accountable, but also just feel there's a certain degree of honour and pride, I think, that exists between us because we know we're acting on behalf of and for our communities.
Paula?
What I like about platforms is that because they are so modular, that word has been used by a number of us tonight as well, it essentially allows us to already start doing work in preparation for something that we do not yet know, right? It sort of sounds like this impossible challenge. How can we put effort or work or science into developing something we don't even know what it needs to look like yet? And so normally that's not how we develop therapeutics. Normally we know the problem and then you reverse engineer, you identify your drug target and then you take that through all your testing. So what I like about the platform technology is that we can already work on the mRNA, understanding it better. We can already work on the delivery methodology and making it better. We can already work on how can we use mRNA to deliver a nanobody, like what Wei Hong is talking about. Or we can already talk to fellow scientists like Ash to learn if he discovers what cell is being wonky, then I can try and tailor my delivery vehicle, my delivery mailman to go to exactly that cell. These are all challenges that we can already solve, even if we don't know yet what the next pandemic will look like. And that is what I like about platforms. And that is why I think it is of essence that we work on platforms to prepare us better for the next pandemic. And because I'm seeing as part of this coming global center how much effort and focus is given to these platforms, I'm very positive that we're going to be placed better than we were before.
And Sharon?
So I might just deal with the second question because I think we've heard about platform technologies really explained
Optimism.
extremely well. Yeah, I'll talk about optimism, and I'm inherently a very optimistic person. However, the world is undergoing tremendous change currently, particularly in response to the pandemic, particularly in relation to infectious diseases, and largely as a result of the change in US administration. And so the US have been the biggest funder in science for decades. And much of what we benefited from in the pandemic with vaccines, therapeutics, diagnostics came from the US. And we're now in an environment where we may no longer have that level of commitment. And I think that's really does worry me. And however, we have got great science here in Australia. We don't want to do this alone. As Jeff says, the most important word in the coming Global Centre for Pandemic Therapeutics is global. And so we want to build a large critical mass of people here in Melbourne so that all these collaborations can happen. They do happen more easily when you're physically right up close to people. But we also really want to partner globally, and we already are doing that. We're funding many investigators across Victoria, across Australia, in the UK, in Germany, and many other places. So science is, when you want to tackle a really big problem, you want the very best people. So we don't want to do it on our own in Melbourne, but in partnership and growing our capacity and capability in multiple places. I think that's a very important goal of ours for resilience against future pandemics.
Yeah, great. So always challenges, but the outlook is optimistic, which is good. So now it's time to hear from you. We'll take some time to field some questions. We do have obviously limited time. I think there's a few roving microphones up the back. So we'll just go through a few questions now. We've got one down the front here. The gentleman, someone can race down with a mic.
Thank you. Hello?
Yep.
Yeah. Thanks. Great session. One comment and one question. Comment, I was living in London during the pandemic and the lectures from Melbourne University about COVID were fantastic. Sharon and the team that did that, I've got to say, it cut through a lot and really gave us a lot of reassurance. And it was, that was fantastic. So communication you can do. You've done it before. The next one, when you've got your platform technology, to me, I worked in drug development and scale up and commercialisation. And what I worry about, I guess, is scale up and then robust supply chain. And I just wonder how you kind of, with so much forward thinking, how you're starting to build those sort of scale up and supply chain strategies for the technologies that you're working on.
So Sharon, do you want to field that one?
Thank you very much for the comment. I should actually acknowledge Kshitij Kapoor, who actually hosted all of those sessions in the early 2020. That he was the dean at the time, and I thank you for the feedback on that when information was hard to get. Well, I think the first thing is we can't do everything as the coming global centre, and we're not a drug company. So we are definitely working in the space of upstream discovery research to build novel technologies. That the goal is that we would need to partner with industry to take us through. We're not naive, out of every single great idea about a drug, 1 in 1,000 might get through, and it takes billions of dollars to develop a drug. That is not where I think the centre cannot do that. But what we can do is develop novel technologies, dream really big and think about, discovery science that will deliver the technologies. Make sure we protect that discovery through very good and aggressive approach to intellectual property. And we've got a lot of capacity in that area at Melbourne University, but we're building more into the coming centre. And then aggressively pursue partnerships. We've got to work with the experts that make drugs. That's not going to be what we do. But it's a really brilliant and important point because this is a tough business for sure.
More questions? Yes, down here we have one.
Thank you. That was wonderfully explained. I loved all the analogies used to explain very, very complex science. I guess my question really is, yes, we need to develop the therapeutics, which I think are really important, but community has to come to the table to take it on. And I think what we've lost as a result of the pandemic is the trust of community. Even COVID vaccinations this year are lower than take-up, way lower than what we needed to be. And how do we actually gain that trust of the community to come forward and support the work that we're doing?
Maybe we'll speak with Paula and Ash, having kind of done a bit of science communication directly to the community through, in this case, some media. How do you feel about tackling that challenge in terms of kind of winning over and bringing the community along for the ride, so to speak?
I think it's a very important question and very important to address this. It's not easy, especially for a lot of fundamental scientists, to envision how their research can accelerate and have an impact and be developed into a therapeutic very quickly. And I think the COVID pandemic has shown that some technologies that were known to the scientific research community were not yet known to the public. And I think that's a gap that we have to bridge. And I think we can play a role there. And I think a big learning that I think a lot of me and my peers are aware of now, that we need to engage and have these conversations much earlier on. Whereas previously, I guess we were all in this idea of, well, there will be time. And when it gets closer to market, or when we have that partnership with an industry, then we'll start and have this discussion. And I think we've learned now that that is too late. And so I think we're trying to, as an institute as a whole and as a coming global center, trying to develop a lot of ways. This podcast series is, of course, one of them, to try and have that conversation earlier on.
Ash, did you have anything to add?
I've got my personal take on it is that as an academic, someone who's always had his head in a book, I really liked my ivory tower very much and didn't really want to pursue any conversation with anyone who wasn't in my ivory tower. That's changed somewhat, and there are two principles that are serving me better now that I've exited my tower. Or maybe I'm still in it, I don't know. One is respect, so I try to list and I try to listen. And there are a few people who, back when the vaccine had arrived in 2021, where it became fractious. People would say, "Oh, just get the bloody vaccine, will you?" And I'm not sure that way of preaching is very helpful. So I think my take on it has been since then to try and listen to people, hear their views, and then maybe they'll listen to my views. So it's respect and listening. That's all I've got.
Wei-Hong, did you have anything you wanted to add in terms of the communication piece and bringing everyone along
I think it's a very important question, and I think for the scientists, it's also reaching out in spaces that are different for us. And so these are spaces that are in family, potentially places of worship, places that are culturally different from our scientific community, and feeling comfortable in actually engaging with the science, and as Ash said, to listen, because the spectrum can be quite wide between someone who believes in vaccination, someone who doesn't. But I think we need to sometimes step back and not speak about the science, because we have to understand that in different cultural backgrounds, experiences with vaccinations can be very different, and our assumption should not be that that is a medicine that is easily taken up. And so I think having that humility to understand that, but I think reaching out in places where community feels comfortable, and then having those conversations is very important.
Great. And I think we've probably got time for one more. If there's one final question anyone has. No? Okay, I might ask one then. In terms of preparing for the next pandemic, and obviously we've discussed a lot here the collaboration that is critical to the success of that. And then Sharon, you also spoke about obviously the collaboration in person is obviously much easier than trying to do so globally. How do you sort of see that, finding that balance, reconciling between collaborating globally while also being quite spread, obviously, and ensuring that the research does progress in a kind of effective and expedited manner?
We've thought about that pretty clearly, and meaning that global collaboration is really important, as is critical mass. And so of the money, the gift that Jeff has given to the Doherty, we plan to spend 30% of it external to the Doherty. And so we are funding and will continue to fund and partner with institutions globally. And we hope to do that with the very best institutions. We have a really exciting partnership with the University of Bonn, who we've worked with for many years. They've got tremendous expertise in the immune response to viruses and ways to develop therapeutics to boost the immune response. And so, we've got a commitment to give 30%, which is pretty unusual, but that's the strategy for ensuring that we are truly global. And when you are willing to fund things, people do want to talk to you. So we've had some really good conversations with some of the best institutions globally. And also now is getting a tough, money. A lot of investment was going out for science through the pandemic. People were very nervous, they were anxious. Governments were spending, philanthropists were giving a lot of funding, but that's changed really quite dramatically in just 5 years. So it's a tough environment out there for funding, and so we want to leverage and partner with other, great scientists around the world. Great.
All right. Well, I'll kind of see that as a end of the Q&A, seeing as I fielded the last one. But it's clear that the next chapter of pandemic preparedness won't just be about reacting faster, it will be about understanding deeper. So whether it's through technologies like spatial transcriptomics, nanobodies, mRNA, or rethinking how we turn homegrown ideas into real-world treatments, innovation is happening right here in the heart of Melbourne's biomedical precinct. We hope tonight's conversation has sparked new ideas, raised fresh questions, and helped build trust in the science that will shape how we respond to the inevitable next pandemic. And of course, this evening was just a teaser. You saw some of the wonderful clips that were extracted from the show itself. To dive deeper into the fascinating world of pandemic therapeutics, check out Off Script, a podcast by the Coming Global Centre, available now on all major podcast platforms. In closing, please take a moment to share your feedback on the event via our short survey, which you can access through the QR code on the screen. And finally, please join me in thanking our panel, Professor Sharon Lewin, Dr. Paula Savall, Associate Professor Ash Hake, and Professor Wei Hong Tam.